Hello and welcome to this plenary session of the Global Young Scientists Summit 2021. My name is Thorsten Wohland. I'm going to be the host for this session. And please note that if you have any questions during the seminar, please use the chat panel which is available to you. So without further ado, let me introduce the speaker for today. And it's my great pleasure to introduce to you Professor W.E. Moerner, the Harry S. Mosher Professor of Chemistry and Professor of Applied Physics at Stanford University. Professor Moerner did his PhD at Cornell University, after which he worked at the IMB Research Division, before becoming Distinguished Professor at the UC San Diego. After that, he joined the faculty of Stanford University. Professor Moerner is a true pioneer in single molecule spectroscopy. He was the first person to detect single molecules in solids. That was documented in a physics review letter paper in 1989. And then he discovered as well the blinking of GFP, the green fluorescent protein. These two achievements were really at the base of what allowed super-resolution microscopy to be developed. And for this, he received many awards, and of course, as well, the 2014 Nobel Prize in Chemistry. So without further ado, Professor Moerner, please, over to you.
Thank you very much for the wonderful introduction. And I also want to, of course, thank the organizers for inviting me to speak. It's a great pleasure. Hello, everyone. This story is about light and single molecules. And so to give you a little roadmap of what I'd like to describe, I'll tell you first about the early history in a few moments, not very long, and then describe three-dimensional super-resolution imaging, where point spread function engineering is used to get axial or Z information to get 3D images. A new type of light sheet then can be invented that shows the power of these point spread function designs to get three-dimensional information in thick cells. I'll talk briefly about looking at the mammalian glycocalyx as a special and very interesting structure that can be observed with super-resolution, and end with correlating electron tomography with a single molecule localization microscopy. So going back to the very beginning, think back to the middle 1980s, and I was working at IBM Research, and we were interested in optical data storage using the color or the wavelength of the recorded bits. And that was a fascinating concept to explore, but looking at the fundamentals of this problem really led us to single molecule detection in 1989. So this first optical detection of single molecules, which was thought not to be possible, occurred for pentacene molecules in paraterphenyl. And this little W shape here is the absorption of a single pentacene molecule. Notice this is a low temperature experiment at the beginning. It was quite a special and fascinating regime, and it required specialized laser FM spectroscopy. But it was an exciting development to get started on being able to detect single molecules. Immediately afterward, Michel Horry detected single molecule absorption by detecting fluorescence, and the field went wild to look at what can be done at low temperatures with spectroscopy of individual molecules. But in the mid-1990s, various people started observing these single molecules at room temperature by a variety of methods. And at room temperature, with detection of fluorescence, molecules are pumped from a ground state to an excited state, and this red-shifted long wavelength emission is what is collected. So imagine you want to observe a protein, and it's not fluorescent by itself. You might have to add a fluorescent label to it, like these small fluorescent labels, which are on the order of a nanometer or so in size. And think about the way you would do the experiment at room temperature. You're really looking at a sample, and you want to make sure that only one molecule is pumped at a time. So you have to place the molecules far apart because the laser spot or the region being probed is never infinitely small due to diffraction. Still, if you do place them far enough apart, you can collect the light from single molecules. So the whole area of studying individual molecules was explored in great detail and still continues to be explored. Here's a quick example from the early 2000s, looking at a transmembrane protein in a cell. We're looking at the surface of the cell, and in this experiment, you can see that at room temperature, these transmembrane proteins are moving all around. There's a fantastic dance of the molecules on the surface. You see that even though they only appear to be, they're only 1 nanometer in size, they appear to be some microns, I'm sorry, a few hundred nanometers in size on this image. And that's coming from that diffraction effect that I just mentioned. So the beautiful thing that occurs when you start exploring the single molecule regime is that surprises also begin to occur. For example, in 1997, we decided to look at individual copies of the green fluorescent protein in bacteria. So in this demonstration, I want to just note that these molecules are not moving. In the previous movie, they were diffusing around, but here, they're fusions to a particular protein that's static, that's not moving around inside these Caulobacter crescentus bacterial cells. A white light image on the left and a fluorescence image on the right can be compared. But the amazing thing is when you start looking at the molecules as a function of time now, remember nothing's moving. What you see in the movie is a fantastic blinking of these single molecules on and off in a random pattern. All of these blinks of the emission from the molecules are coming from inside these cells. And this was another surprise. It's coming from interesting dynamics and dark states of the fluorophore of fluorescent protein, but it turns out to be something that maybe some people thought was useless. Yet, in fact, this is one of the key components of super-resolution optical microscopy. So, how do we circumvent that optical diffraction limit? I'll describe that now in a moment. Imagine you have, again, a bacterial cell, quite small, just a few microns in length, and a particular protein has been labeled with a fluorescent protein, and you want to see the structure. Someone might think that all you have to do is to buy the most expensive optical microscope possible, and then you'll see all of the detail. But in fact, that's not true because of the diffraction effect that I just described. You see a blurry and fundamentally fuzzy image because the individual emitters, even though they're only a few nanometers in size, they appear to be a few hundred nanometers in size due to the diffraction limit first mentioned by Ernst Abbe in the late 1800s. Roughly lambda over 2, the wavelength divided by 2, is this approximate size of even a point emitter would look that big. So that's a diffraction limited image. And using, on the other hand, by using super resolution microscopy methods, this image becomes that image. In this particular case is 5 times beyond the diffraction limit. And you see a great deal of additional detail that wasn't possible. A 5-fold improvement in resolution is not a small effect. And it's because of this breakthrough, getting beyond the diffraction limit, that there's much interest in super-resolution microscopy. Now, Stefan Hell used a method called stimulated emission depletion microscopy to achieve this. Eric Betzig and I utilized a single molecule method. So let's describe how that works. How can you surpass that diffraction limit using single molecules? So the first thing you have to do is to recognize that if a molecule is separated from all its neighbors, the image of a molecule on a pixelated camera can be fit with a function that allows you to find the position of that single molecule to very high precision, far beyond the width of this diffraction limited spot. So that is single molecule localization. And the ability to do this improves depending upon more photons being detected to define the shape of the point spread function. The next thing you need is some active control mechanisms to make sure that the number of molecules emitting is at a very low level. You want to think about having molecules that have an on state and an off state. Even though you're shining light on this off state, you don't see emission. But if it's in the on state, when you shine light on the molecule, you see fluorescence. So there's many ways to do this. There's many ways to achieve this on-off process. Some of them are photoactivated microscopy or blinking, like I showed with the fluorescent proteins, or other types of photoreduction, as in the STORM methods. If you have that, then here's what you can do. Suppose you have a structure that actually has these ring-like shapes. If you put fluorophores all along these rings, if they are all on at the same time, you just see this blurry image, because these spots, which we call the point spread functions from individual molecules, overlap badly. And that's the fundamental problem with doing diffraction limited imaging. But if you have a control mechanism, then you can make sure that only a few are on in a given frame of a movie. And you can localize those in that frame of a movie. Going to the next frame of the movie, if they're blinking dramatically, different molecules will turn on, and so you'll get new samples of the underlying structure. You save all of these positions of where the single molecules were found, and then show them all at once. And now you see that you get information and structure beyond the diffraction limit in a pointillist sort of way, just like pointillist art. So this has been called PALM and STORM and many, many other names. This acronym here simply emphasizes that you need to see single molecules and you have to actively control their concentration to make this work. Okay, so here's a real example now in real cells. And this is a case where there's blinking fluorophores. So on the left, I'm showing you the diffraction limited image of a cell now, a eukaryotic cell. Here's a scale bar, and this happens to be tubulin, immunolabeled with a dye, Alexaflor 647, which is one of these blinking dyes if reductants are added to the solution. So in this one frame, you see single molecules, and on the right, you also see the positions of those single molecules from that one frame. But remember, they're blinking dramatically, so in the next frame of the movie, more molecules turn on, the earlier ones turn off, and now you get more positions on the structure. This is a fixed cell, so that things aren't moving while all of this is happening. Next frame, more locations, next frame, more locations, and throughout the movie, all of these blinking single molecules provide this fantastic information at high resolution beyond the diffraction limit of the actual structure that had been labeled. So this idea now, of course, has been applied in many, many laboratories to look more deeply at many, many interesting systems, from material science to biology and so on. Here's just a very small number of examples just to illustrate. This happens to be an axon, and the protein being labeled is spectrin. And so the Zhuang lab showed and discovered that these proteins are organized in lines perpendicular to the long axis of the axon. A fascinating bit of structure that's beyond the diffraction limit, not seen before super resolution microscopy. This happens to be the synaptonemal complex done in super resolution, but also in 3 dimensions. And I'll talk more about 3 dimensions in a few moments. Here's an example from amyloid diseases, the Huntington's disease in particular. These are labeled Huntington proteins of the mutant form, and they form these fascinating, if you like, little fibrils close to, in this case, a very, very large inclusion body. Again, requiring super resolution to be observed. The last example I'm showing you is the glycocalyx, the layer on the surface of cells that is covered with glycans. Here they're forming tubules, and I'll talk more about these tubules and the glycocalyx in a few moments. So now continuing with this, the power of these super resolution experiments, let's talk about 3 dimensions for a few moments, because you know that the world is 3-dimensional. Even a small bacterium, if I'm thinking of the motion of a transmembrane protein on the surface, in one of the membranes of the cell, If you only see its projection into 2D, you will make mistakes on its diffusion properties if you can't see 3D. So there's a great interest in three-dimensional imaging. There are a number of ways to achieve three-dimensional imaging. The way that we do it in my lab is to change the microscope in a fundamental way. So please look at this little illustration of the key features of a standard wide field microscope. The light from a molecule is collected and then goes through the microscope. A tube lens forms the final image on the camera. But what we've been doing to get three-dimensional information is to place something special here in the pupil plane or the Fourier plane of the microscope, this phase pattern, which shows you how light is delayed at different positions in this pupil plane. The effect of this fascinating pattern is that the light from a single molecule appears as two spots on the camera, not one, but two spots. More importantly, if you think of now placing the molecule at different axial positions, watch what happens. These two spots revolve around one another. So that if I have a molecule, let's say at that Z position, a particular Z position, then a line through these two spots and a calibration would let you determine what Z position it is located at. So that is the way in which three-dimensional information is encoded into a two-dimensional camera. The midpoint between the 2 points, of course, can be X and Y, and Z comes from the angle of the two lobes. So this idea is called the double helix point spread function, and let me emphasize a couple of important things. First of all, there is no scanning in this type of three-dimensional imaging. All the molecules in a thick slice of the sample, a few microns in thickness, appear at the same time in the frame. And you can clearly see that there are molecules with different angles between those two lobes. That means they're at different Z positions. Another key thing to remember is this microscope normally would have only a few hundred nanometers, let's say 500 nanometers of axial range, but using this point spread function, the axial range increases to 2 to 3 microns. Now that's an interesting feature of these special point spread functions that we're going to utilize in a few more moments. Anyway, if you now apply this imaging protocol to blinking molecules or molecules that are photoactivated that produce super resolution information by active control of their concentration, you can get three-dimensional images in here in 2 colors. In this case, it's bacteria. On the surface of bacteria is one label, and inside the bacterium is another label, a different protein that's been labeled with blinking fluorescent proteins. So the interest in this technique, of course, needs to go beyond just this short Z range of a bacterial cell, which is only about 500 nanometers thick in the Z direction. So let's talk for a moment now about thicker cells. What do you do when the cell is thick? It's already well known that a light sheet can be very powerful for thick cells. This is showing a light sheet going through a thick cell from the side. So we're looking at a pancake edge on. And that has the useful property that molecules above and below the sheet are not pumped and therefore not photobleached or anything else. But of course, a microscope is looking at the emission from this sheet of irradiated sample normally, and a lot of people have worked on these light sheets. But there's a little bit of a challenge. If you have a cell, let's say adherent to a surface, and you want to look down at the bottom of the cell, then the light sheet has to go through the corner of some sort of, for example, a chamber that might have a buffer and so on around the cell. And so in order to avoid that difficulty, lots of designs have included some complexities, custom-made parts, different objectives in the fluid, and all sorts of things to try to address this issue. So we realized that with these point spread functions, you can solve many of these difficulties simply by tilting the light sheet, tilting it and sending it to a sample at an angle, just a few degrees, about 10 degrees is all that's necessary. The thing you might be worrying about now is all of the fluorophores that get pumped by the light sheet are, of course, above and below the focal plane. So how is this going to work? How am I going to be able to image them all at the same time? Well, that's solved by these point spread functions I just described, because they have a large axial range. So we implemented this idea and call it Tilt 3D for a tilted light sheet microscopy. You can see that the illumination is trivial. It's simply a cylindrical lens producing a standard sheet of light that is maybe 1 to 2 microns thick in the sample, because there's no particular need to make the light sheet extremely thin. As long as you have a lower concentration of emitters. We are able to, throughout that 2 micron thick layer, detect the X, Y, and Z position by using a double helix point spread function in this simple 4F optical processing system. A second point spread function called the tetrapod is also used in these experiments because it has a much larger Z range and allows us to measure the position of a fiducial for calibration purposes. So this idea was worked on in my lab by a postdoc, Anna Karin Gustafsson, to implement the idea. And here's an example of looking at mitochondria, where TOM20 has been labeled in the mitochondria. And you can clearly see in the XY, XZ, and YZ projections the interior of the mitochondria. But again, this particular structure it doesn't extend over such a large Z range. Okay, let's have this move. It's not moving that well. That's okay. So we can illustrate that the idea also works easily for thick structures, such as, for example, the nuclear lamina, which is this sac that sits just inside the nuclear membrane of a eukaryotic cell. So this is, again, storm or destorm used to make fluorophores blink that are labeling the lamina, lamin B1 proteins. The double helix is being used in each position of the light sheet, and the fiducial is explored by this tetrapod for calibration purposes. So this particular 3D image is quite beautiful, and you can see that there is an intranuclear channel through the lamina going from one side to another, and other dimples in the lamina and so on. That is for a structure that extends over 5 microns in the Z direction. So that's one of the neat things that have been happening in 3-dimensional microscopy. And let me just note that these approaches are better than confocal microscopy because the resolution is far higher, and the wide field imaging allows you to look at many molecules at the same time. So it's in principle also faster for recording the information from a thick cell. Well, not only mitochondria, but also nuclear lamina. And this goes back to that glycocalyx that I mentioned earlier, that we can also see those tubular structures that extend out from the surface of a cell. And I'm going to talk a little bit more about this structure called the glycocalyx now. Switching to 2D, because there's so much interesting happening in 2D. This team of people, both in my laboratory and in Carolyn Bertozzi's laboratory, took a look at the glycocalyx, this covering on the outside of the cell that's composed of glycans and glycoproteins that are heavily involved in communication between cells, but also are heavily elaborated in cancer systems, cancer situations. This cell line is a breast cancer cell line. So let's look at some super resolution images of those BT20 cells. You can clearly see a nucleus here, and remember it's a 2D slice, so that you can see very clearly for these tubules that are present on the surface that the super resolution microscopy was able to show the interior of these tubules. And because of that, we can measure the width of the tubule and try to get information about the thickness of the glycocalyx. Of course, this kind of detail is not present in the diffraction limited imaging that I'm illustrating over on the right. So this is another great example of super resolution letting you see more, which can be applied to both normal cells, but also cells that have a pathology. So in this study of the glycocalyx, please remember that the glycocalyx is this very complex forest of interesting glycoproteins and glycan structures on the outside of the cell, attached outside the membrane. We're mostly going to measure the width of the cell from side to side by fluorescent labels that are attached to specific glycans, either the GalNAs label or the sialic acid label, so that we can record these super resolution images for one type of glycan or a different kind of glycan. The sialic acid extends further out, the GalNAs is closer in. But more importantly, you can observe these structures for different kinds of interesting cancer cells. These are cancer cells that came from patients at a primary site of a tumor, in this case, an ovarian cancer tumor. The width of the cell, remember I talked about width, from lipid to lipid is smaller than the extra width that comes from the GalNAs labeling and the extra width that comes from the sialic acid labeling. This works for several situations, also in metastatic cells. In other experiments that I don't have time to describe, where the RAS signaling pathway could be turned on, we showed that the thickness of the glycocalyx increased, the height of the glycocalyx increases under those situations, leading to tumor progression. So it's fascinating that something about the elaboration of these glycocalyx thicknesses is involved with the cancer progression. Lots more to study in that system, but time moves on. So let me now turn to another interesting new development that we've been able to explore in recent work. I want you now to back up and think about both super resolution optical microscopy, but also electron microscopy, which is extremely exciting these days. And this is a great drawing from David Goodsell. I'm describing here the work of Peter Dahlberg, postdoc in my lab. But of course, as you know, these cells, in this case a bacterial cell, are filled with so many interesting structures, both DNA, nucleotides, and proteins, and lipids. And here's that the surface that I was talking about. And so you would like to recognize that there are, of course, ways to observe this with extremely high resolution, like electron microscopy. Our super resolution microscopies get down to, let's say, 10 nanometers or so. Electron microscopy goes much further, but each of these methods has strengths and weaknesses. Let's think about cryogenic electron tomography. Okay, for example, you can get an image that has very, very high resolution now, down to 1 nanometer and below with CET. However, what you see is a grayscale image. You see blobs, and you might not know which protein is which. So it gives you high resolution and the general context of the cell, but it doesn't give you the exact identities. On the other hand, single molecule fluorescence involves labeling a specific protein by a fusion, for example. And so now in our optical imaging, the light comes only from those proteins. So there's great specificity and of course single molecule sensitivity, but not as much context from these kinds of imaging. You of course can do a couple of colors, but that's not quite as much as you would like to see. You'd like to know where these are in the context of the cell. And so we've been highly motivated, because if you can do this, then you know that these spots from the optical microscopy come from these particular proteins in the CET image. So in these experiments, we have to, of course, do the work on a sample that's already prepared for electron microscopy. That is a sample that's frozen on an electron microscope grid. So this requires microscopy at low temperatures, first for single molecule localizations and then electron microscopy. So let me give you a little more detail about that in these remaining moments. First of all, here's a cryo electron microscopy image. It's actually one slice of a tomogram, so let me be more precise about that. This is a bacterial cell again, like those Caulobacter cells I showed you earlier. Now we want to ask ourselves, okay, which protein is which in this image? Well, it's pretty clear that these black dots are the ribosomes. Those are, of course, huge, and so they're fairly easy to pick out. You can also easily pick out a region where there's no ribosomes. And this is an interesting property of Caulobacter, that there is a ribosome exclusion region near the end, where there are other interesting proteins and fantastic details that come from how the pole is organized. That's kind of one of the key organizing centers of the whole bacterium. You can also identify in this picture objects that are periodic or continuous, like, for example, the S layer, which is this outer layer of the cell. There's an outer membrane, then there's a peptidoglycan layer next, and then there's an inner membrane. So that's all fine, and those things can be identified. And what's usually done is to annotate them by hand, to draw in by hand, or even use a computer program to light up the objects that you recognize. But what about the other thousands of proteins that are present inside this cell? Where are they located? So that's the reason that we're pursuing this work of doing optical microscopy correlated with electron microscopy. Now, let's first talk about the optical microscopy. Again, on these Caulobacter crescentus cells, this is a white light image of a microscope grid, and you can see the holes in an electron microscope grid in that white light image, and you can also see several bacterial cells that are highlighted there. But now that the sample is at low temperatures, we're going to do optical microscopy on a particular photoactivatable fluorescent protein called PAMK8, one that we found to be photoactivatable at low temperatures. And you might have noticed there's a few little red spots here at the end of the cell. We're labeling POP-Z, which is one of those proteins in that polar region in this experiment. So blowing up the image even more, you see there's some more detail that is found from the optical microscopy. And then finally, this is a special way of displaying the localizations we found for the single fluorescent protein fusions. The histogram is just simply putting molecule localizations into boxes, but the rings are single molecules. And we're showing the localization precision of one of those single molecules by the diameter of the ring. There's one ring right here in the center that's extremely tiny, so tiny that its localization precision is as small as less than 1 nanometer. Now, why is that? Well, it's because at low temperatures, you get many, many more photons per emitter than at room temperature. That means there's less photobleaching, so more photons before a molecule turns off. In fact, a challenge in this work is that sometimes we would like the molecules to turn off and not just keep emitting forever, because the experiment can take longer and longer as the molecules emit longer and longer. There's a bunch of fascinating physical properties that can be explored here to get new fluorophores. But let's talk about the correlation now between those low temperature single molecule measurements that's been done by my postdoc Peter Dahlberg and others in my lab. What you do is to label the cells in the usual way by fluorescent protein fusions, and then plunge freeze a solution of cells on an electron microscope grid. The optical microscopy is done in a microscope that works at 77 Kelvin. The sample is held at 77 K. Then that 77 K sample is carried to one of these electron microscopes where the microscope is used for low mag and high mag. And then there is a process of image registration and visualization to combine the two images. We call this method correlative imaging by annotation with single molecules, or CHASM. That's how we're pronouncing this, CHASM. And I want to emphasize that we're not doing a complete super resolution image of every protein that might be on a structure. In fact, we're observing a selection of the single molecules, but nevertheless, each single molecule does represent a location of one of the proteins of interest. So let me illustrate that in the case of Caulobacter crescentus again. First of all, let's do a control protein called MCPA, which is located in the chemoreceptor array. This is a structure, the chemoreceptor array, that you can observe in the electron microscopy. You see these lines which represent slices through plates. These proteins are localized on a plate close to the cell membrane. And indeed, the MCPA fluorescent protein fusions, the single molecules, do appear in the region where they're supposed to in this kind of image, because you know where they should be in this particular case. But for many other proteins that are not discernible in the cryo-electronic tomography, such as POP-Z, that's the one that's in the polar region, or SPAM-X, which is another protein that is known to be near the pole, we find single molecule localizations scattered around in several different positions. So the beauty here is that for different proteins that were not observable in an electron microscope, now we can show where they are with high precision, down to, on the order of, in this case, most of them are more like, let's say, 5 nanometer localization precision. So let me just quickly end, because I know that I'm basically out of time, but I want to give some thoughts to you, young scientists, it's so wonderful that you're working on science. Of course, you know that science can be hard work. And so I want to encourage you to continue to be persistent and methodical in your work. It is really important for you to also teach others, younger people, that they should be asking how things work too. They should be asking how their cell phone works, how the world works. That's the beauty of science, to ask these questions. But to you as scientists, remember to push beyond conventional wisdom and question assumptions. Keep your eyes open for surprises, okay? Those blinking single molecules we might have thrown away if we weren't excited about that surprise. And moreover, let me emphasize that there will be failures, but you should embrace failure, because failure means that you have something new to learn. Try to think of what you can learn from that failure. What aspect of the experiment did you not fully understand, and therefore you want to learn more about? It's much better to treat failure in this positive way than to be disappointed by it, or hide it, or not tell anyone about it. So it's very important part of science, which we do because this is a rational and predictive way to understand our world. I want to thank my wonderful past students, postdocs, and collaborators, and the current Guacamole team, we call it. Here's the funding agencies, and a quick disclaimer. Here's a little logo for no ensemble averaging for single molecule experiments. Thank you for your attention.
Thank you very much for a fascinating talk. So we had quite a number of questions, actually, and I'll try to, some of them paraphrase and combine. So the first question was actually by Isabel Kunitza and Christopher Leonard. And they ask, how do you know where a big molecule actually is if it has a single fluorescent label? And how can you make a difference between off-site effects if you're actually looking for ligand binding, for example? Because the label itself is actually relatively large.
So that's a great question. Thank you. These labels are on the order of 3 or let's say to 5 nanometers in size for the fluorescent proteins. But for the glycocalyx example that I gave you, the label is incredibly small. It's extremely small, roughly 1 nanometer, because that single fluorophore is reacted directly to the glycan. So the point is, there are some labels that are, let's say, 3 nanometers in size, and there are some that are even smaller, just a single fluorophore. So you're correct that these experiments are showing the position of the fluorophore, which may be 3 nanometers to 5 nanometers displaced from the protein of interest that you're caring about. So remember that our localization precision, of course, is still a little bit larger than that, some 5 nanometers or 10 nanometers. In normal super resolution, the low temperature one is doing better, but you're absolutely right that there may be a need to recognize that the protein of interest is slightly displaced from the localization that we find. Nevertheless, this entire technique gives a lot more information about where the protein might be in that complex CET image than you have otherwise.
So before I go to the next question, I just want to let you know that there's a lot of actually comments of people actually who were very happy about the inspiration and the advice you gave in the end. So,
thank you.
So let me go to the next question. That's actually a very interesting one. That was by Julia Emilia Brumboya. And asked, is there any way to obtain a super resolution image of a moving nano object using blinking single molecules?
So when you have a moving object, there is, of course, a rate associated with that motion. And in this type of super resolution that I've described, based on, let's say, blinking, there is a certain time period required to get enough images from a camera to get enough information about where the single molecules are located. So it turns out that this is really a trade-off. If you want the molecules to blink faster, you can work on increasing the intensity, usually, because most blinking processes are light-driven. And so some groups, like Jörg Bjørsdorf, for example, has worked to increase the blinking rate dramatically, and therefore having such a fast blinking rate with such a fast frame rate camera that they can see motion at video frame rates. So this is one of the trade-offs that you have to choose in a given experiment that, of course, is also affected by the field that you might want to look at, the field of view, and so on.
The next question is by Dominika Dorowicz-Powa. Sorry. And the question is actually about machine learning. Is there any attempts to try to use machine learning for single molecule localization?
Yes, that's a wonderful question. Thank you very much. And indeed, the answer is yes, yes, and yes. There are many groups who have proposed and published different algorithms for single molecule localizations. You can use it to find the positions of the molecules faster. You can also use it in a fascinating recent project that we worked on, actually, called background estimation. My postdoc Leonhard Merkel created a specific neural net that has the property of extracting the background from an image. So this is really fascinating in the following way. In a lot of our experiments, we of course have background. We have background photons, but it may be structured. And in a cell, if the background is structured, of course, then it can confuse your ability to find single molecules precisely. So you'd like to estimate the background and remove it as quickly or as easily as possible. So this neural net is trained on the shapes of the single molecule point spread functions in both 2D cases and in 3D cases. It works for both. But the idea is the net is told what the good data is, which is the point spread functions that you're after. And so it takes the rest, and that becomes the background. So it estimates and provides the background, a structured background that can be subtracted from the image before you do localization. So that's just one of the things that's been done with neural nets. Other people like Yoav Schachtman has worked on inventing a neural net that will itself calculate and produce new point spread functions for particular applications, and so on. So it's a wonderful growing area.
The next question was actually asked in different ways by multiple people. That's by Kajana Bunpalita, by Bernadette Lesella, and Ananya Biswasa. And the question is, how can you control the turn off and turn on of molecules, and how does this actually influence your photon rate, which you need to get the super resolution?
So it's a great question. These are fascinating photophysical parameters, if you like, and photochemical parameters of the fluorophore itself, which is why we have to understand these fluorophores as deeply as possible and invent new ones with slightly different properties where necessary. The key point, let's say for blinking, is that the molecule enters a dark state. And so the rates at which it enters the dark state can depend upon triplet yields and can depend upon the concentrations of these reductants that are added in the storm and destorm processes. So you can change things somewhat by changing those concentrations of those reductants, or changing the molecule itself in terms of its intersystem crossing rates and its triplet lifetimes or its dark state lifetimes. In the case of photoactivation, the molecule may turn on but eventually photobleach, and so the total photons that you get depends upon these parameters as well. So a deep understanding of the photochemistry and photophysics of the molecular energy levels is what is needed to understand these properties. Of course, the intensity is also a key variable that can be used to change it. Other colors can be used to turn a molecule on or off, perhaps. So we can imagine using a number of different tools that you might have to affect these blinking rates and on times and off times for the different molecules.
The next question is by Jake Thomas. Jake Tomasa. And his question is whether you think that imaging cells on glass or in the frozen state changes their biology, or if this is actually biologically relevant.
So again, that's a wonderful question. There will certainly be some cells that don't like to be on that surface. And so there's a number of people who are looking at single molecules in more interesting complex matrices that mimic a more biological matrix. There's even some experiments that have been done in organisms like worms and so on. So I think the deeper way to think about your question is that if you saw some strange behavior, you might want to try to replicate it in different situations that might have softer surfaces or might have different kinds of surfaces to see if you see the same behavior. There's, of course, a whole area of nanobiology where small structures are being used to maybe affect cells in different sorts of ways. And that's another example of an interesting case where you might want to explore the science of what might be happening caused by the local surface near the cell. So it's a good question, a good point, and worth always thinking about, depending upon what level you want to understand that biology.
The next question is by Antoine de Saint-Germain, and he's asking if the Abbe diffraction limit is your limit for light microscopy. Is there an equivalent for nanoscopy?
Nanoscopy is kind of a big term. If you mean electron microscopy, the electron microscope is based on using electrons, which have a very short de Broglie wavelength. And so the de Broglie wavelength can be small down into the sub nanometer angstrom scale and so on, depends upon energies and that sort of thing. And of course, people want to go to higher and higher resolution, but that entire technology is somewhat limited ultimately by damage to the structure. If you have too high of an energy or too long of an irradiation, the electrons ultimately damage. So, fundamental limits is always a term that people throw around a lot. In the case of the Abbe diffraction limit, that is, of course, a fundamental limit for far field diffraction limited microscopy, which has been broken by these various techniques of STED and single molecules, and this new scheme called MinFlux that has a lot of fascinating behaviors as well. It's another scheme popularized by Stefan Hell. And so in each of these cases, and usually what happens is that some technical limit finally appears that makes things sort of stop at some level where you can't go any further, and might need to think of a different way to do the experiment.
So let me ask a couple more questions, because I thought they were very fascinating. I mean, one was by Harold Matthew Pan and Sunku Srivatsava. And the question is, can you do super resolution microscopy in live cells? And can you use it perhaps even in point-of-care diagnostics?
So a great question. Yes, we actually have done a lot of super resolution microscopy in live bacterial cells. The situation is the following. Again, there was an earlier question about time scales. Some structures are quite stable over long times, that they're part of a skeletal network or something like that. And so you can do these experiments in those cells. Of course, there may be lots of other molecules and proteins moving around, but the ones you're interested in are not moving so fast so that you can image them. The basic idea is that live cells ultimately may be damaged by extremely high intensities, but a lot of the cases where these measurements work well, almost always I find to be two-dimensional structures in the three-dimensional object, which reduces the number of fluorophores that might need to be pumped and recorded. in an extended imaging experiment in live cells. And the other case you mentioned was in organisms. Is that what you said?
Point of care detection.
Point of care. Well, absolutely. This technique can definitely be used in principle in point of care. Always the issue is, do you have an assay, and what does the assay tell you? What do you need to image that assay, let's say? And so clearly these components, the cameras, lasers, and so forth, can be miniaturized pretty well, so that you can imagine doing an image. The question is, what about that image is going to tell you what you need to know? Maybe to throw out an example, something that you might want to image regularly, might be malaria parasites, or might be other sorts of structures that are important for certain diseases, and you want to see something in very fine detail. So you could imagine that it can be done, yes.
So there were many questions, and I can't ask all of them. So let me end this session, though, with a more lighthearted question, but as well quite fundamental. So Tama Yamagoshi is asking, among all what you've ever seen by using your devices, what impressed you the most?
What structure impressed me the most? Is that what you think is being asked? What structure? Or
I think what she's asking is, like, what was the most fascinating thing you saw with your techniques?
Okay, well, let me just say that the process of discovery, of course, involves a surprise. And for me, probably one of the most exciting things was when we saw this blinking and switching phenomenon at low temperatures. At low temperatures, a single molecule is maybe in a crystal or in a polymer at 2 degrees Kelvin. And so it has a very, very sharp, narrow absorption line. And so at those temperatures, of course, there's no motion, essentially, right? But in fact, the molecules were jumping back and forth, okay, in frequency space, which was the big surprise that occurred back in 1990 when Patrick Ambrose, my postdoc, came running into my office and he says, the molecules are jumping around. And this was, of course, a wonderful moment, a truly great moment, because it's something we just simply did not expect in that crystal. That was really a very, very exciting moment, because when you see something like that, you say, oh, wow, we have to explore it. We have to try to understand it. We have to think of what we can do with it. We have to all sorts of things. Anytime that kind of a discovery occurs, a surprise, you don't know how it really might be used in the end, decades later. And that's the amazing aspect of this kind of science, that the spread of people working in this area over the whole world, the new things that were done, the learning more and more about each of these physical processes ultimately led to some fascinating applications. And I think there's a lot more to do, because single molecules really are an elemental object, if you like, right? An ultimate object. What can we learn by watching those individuals and seeing what they tell us?
So with these words, thank you very much for a really fascinating lecture and inspiration for many of the people in the audience. Then I'd like to thank as well the audience for joining and listening. And then I hope to see you in the other plenary sessions. Thank you very much.